Hello, my name is Dr. Annie Kathuria, and welcome to our podcast, Conversations with an Organ Engineer. Today we discuss what autism spectrum disorder is and its neurodevelopmental biology within patients. We highlight current tools such as organoids and discuss what the world of tissue engineering can mean for curing autism.
Hi guys, welcome to a conversation with an organ engineer. Today we have Dr. Annie Kathuria and I am Ananya Shirsagar, a current student at Dr. Annie Kathuria's lab, working since 8 months now. Currently I'm a master's student studying masters in biotechnology at Johns Hopkins University. So today's topic is autism spectrum disorder. So the first question I have is a very generic question as to what is your definition of autism spectrum disorder?
So autism in general is characterized by three main characteristics. It doesn't have a particular blood test or a biomarker like diabetes would have. It's diagnosed by a child psychiatrist or a pediatric family doctors or someone that is close to the family. Usually it's based on the definition that the kid needs to meet these three criteria, which is lack in social communication. So they have an inherent ability of not being able to communicate as effectively as a normal child would be. They have obsessive compulsion behaviors that is noted, and some have motor and cognitive deficits. So these are the three combined. Some may not have cognitive deficits, some may have it. So this is like the combined diagnosis that is done. And that's why the term autism has been now changed to autism spectrum disorder, because it now carries a variety of autism related disorders, and some it's like a spectrum, it's a scale. Some could be very mildly autistic, so some social communications issues and some cognitive issues, but some may be on very high cognitive spectrum issues, no issues at all, but the social communications issues are really high. So that's why there is now a scale for this, and it's called autism spectrum disorder. So that's how I would define it.
Is there, what was your motivation or your personal experience that led you to study autism during your PhD?
That's a fun conversation. So I was in India. I was 14, 15 years old. My cousin was my little cousin. He was about 8 or 9 at that point. I don't remember his exact age, but he had developmental issues, and nobody in the family could picture it, but I could see it. And he was diagnosed with autism, and it's no particular gene mutation, but he was diagnosed with autism. He had severe developmental issues, cognitive, social, and obsessive. So he was severe autistic case, and that was the trigger. Why is he different versus my little and he had a sister who was completely normal. So why is she normal and he's different, and they're coming from the same parents? So that was the kind of question that intrigued me at that point. And so was very interested, and I wanted to help the kid. I couldn't at that point, and there was no medication for him. They did a genetic test, even that also gave nothing. Mm-hmm. So there was no solution to the problem. And then, being a scientist, at least in my head, being a curious person, why don't we have a solution for this problem? And the more I got into it, and my love for science got all of this into the place. So that's why I actually did genetics and cell biology and development when I was studying in University of Minnesota. And there I learned genetics is half the part. I need to learn more about neuroscience. Mm-hmm. So I did a double major in neuroscience. And then we got all together and tried to solve the problem. Haven't still solved it yet, But I have started developing tools to solve it, and that's where our brain organoids or brain organ-like structures come in.
Okay, so because now we're talking about kids who are involved, majorly suffer from autism, to get their cells, I'm sure there would be some ethical considerations and concerns that scientists have to pass or combat. So how does one go about making sure that whatever studies you're doing are ethically right?
So there are institutional review boards that are established for any stem cell or any patient sample that we get. We have to follow the institutional review boards. They are internal and external. First of all, we don't know what the patient is. When we get our samples, we only get the diagnosis and their age and sex. We do not get who they are, what they do, where they're coming from. We just get an identification number, which is random, like a barcode, with their age, sex, and what the diagnosis was, and what medication if they are on, if we want to divide the different patient types. That's all that we get. We cannot trace it back. So that's one of the biggest roles for us scientists or bioengineers that has been established. But besides that, yes, the clinicians have access to it, and we work with clinicians all the time. I work with pediatrics department. I work with the psychiatric department. We work with epidemiologist department. So we work with a lot of clinical and clinical related, and they have another layer. So this is our institutional review board. They have another institutional review board themselves for their patient collection. So there's 2 to 3 layers of these reviews that we have to go through. Lots of paperwork that we have to do, but these are good things that have to be taken care of. So we do not do any of the patient collection. The clinician does, and they have their own institutional review boards, and then we give the option to the patient if they want to donate or not. So it's entirely up to the patients. And most patients, in my experience, do donate because everybody wants to get their child better. Mm-hmm. And that's what we are here all sitting for, is getting children healthy.
So there are, like, for a student who's just starting to understand what neuroscience is, so there are a lot of terms like neurodegenerative diseases or neurodevelopmental diseases, mental illnesses. So how do you define these categories, and autism spectrum disorder would fall into which of these?
So you are right. So neuroscience or neurological disorders are widely divided into neurodegenerative and neurodevelopmental disorders. For neurodevelopmental disorders, most disorders that happen during early childhood, even before childhood. So some may develop in utero when the mom is pregnant, to up to a certain age of the child, which is young adult. And that are defined as neurodevelopmental disorders because that happens during the development phase of the brain when it develops. And neurodegenerative disorder are disorders that are usually defined after your brain is considered an adult, which is after 25 years old. And those usually happen when you are at a older age. So from your middle age, let's say 40 and above, I would define it as neurodegenerative disorders. And that 40 is also pretty young, but we can say, like, some people experience Alzheimer's and Parkinson's early because of some kind of mutation in their gene makeup, and those can be said 40 and above can be defined as neurodegenerative. Both are very distinct. Both have similarities as well. So distinct in symptomology, similarities in the cellular molecular makeup of these disorders. And now for autism spectrum disorder, as I defined it, it happens during, or most are diagnosed during early childhood. So I would define it as a neurodevelopmental disorder.
Okay, so since all these disorders are in some sense overlapping in terms of cellular and molecular phenotypes, or even in terms of the symptoms that the patients show, if we have to diagnose autism, say with some other disorder like schizophrenia, how do you specifically diagnose that, okay, patient A has one and patient B has the other?
For neurodevelopmental disorders, autism and other autism related or even young adult disorders like schizophrenia are defined by the psychiatrist themselves. As I mentioned, it doesn't have a particular biomarker of sorts. You can have a gene mutation that causes autism. Rare schizophrenia doesn't have any particular gene mutation, but that is also rare. So it's a combination of your doctor, family doctor mostly. For kids, it would be a pediatric doctor who would just like, okay, this kid is showing developmental delays. What could be the reason for it? It could be social, it could be cognitive, whichever it is. And so they will do a genetic test, and sometimes the genetic tests don't reveal a particular gene mutation, but the characteristics are all for autism, like social awkwardness and obsessive behavior and the other parameters, and then they would define it as autism. But as I mentioned, there's no particular marker, like a test, like a blood test. Okay, this is high, this is low, and this is what you have. So there is no particular biomarker.
So do you think this is where the research in autism is lacking?
Yes. This is one of the primary reasons where the research is lacking. There are 2 or 3. One is there is no biomarker for neurodevelopmental disorders. Most neurological disorders have very low understanding, or even we don't even know when they happen, how they happen. So we don't have any biomarkers for any neurological disorders. So it's not just neurodevelopment. And then the doctor diagnoses them based on the symptoms they show, and the excellence of the doctor and the experience of the doctor is usually what we get to once it's defined. And the second part of it is, since we don't have a biomarker, the rate of finding out what is wrong with your kid becomes lower and lower. So the disease might be more prevalent or might be less prevalent. They might be overdiagnosed or underdiagnosed. So that's another issue. And the third issue is, and this is related to our research that we do in the lab, is there's no drugs for it. The drugs that are used are very generic drugs that people have been using for neurological disorders all the time. And so for finding drugs, we usually use animal models. And as far as I know, there are no animal models that get autism. Animals don't get autism. It's a higher cognitive disorder. Schizophrenia, mental illness kind of disorders, and other neurodevelopmental disorders, unless you induce the gene that is going wrong, or I don't think there is an autistic mice out there. I'd be surprised if there is. Unless you induce it in the mice, I don't think it's gonna happen. So this is where our organ technology comes in.
Okay, so the organoids that we are building in our lab, are those relatively a better tool to study this order than the animal models?
Yes. So because we can derive the organoids and the brain models using the patient's cells, as we discussed in our first podcast, the Induced Pluripotent Stem Cell podcast, go back to it and check it out. But yes, we can definitely use patients' stem cells to make the organoid or the little brain in a dish, to understand when the disease happens, how it happens. We can capture cellular molecularly at least and track the developmental trajectory of the cellular molecular development of the brain and the subtypes of brain cells to understand what goes wrong. And if we understand what goes wrong cellular molecularly, we can treat it. We can use the drugs that are available in the market, which are thousands of those, and we can put that in and then see how it rescues those cellular molecular phenotypes. And if it does, then we have a potential drug to give back to the patient, which goes back to our personalized medicine concept. But this is a better model, though the lacking of this model is we have cells in a dish, and those cells in a dish don't have a particular behavior. So I don't know if this particular cell will affect behavior, or we can guess based on the cellular function, or will it affect more cognitively? So that's where the system lacks. It doesn't know how to behave. We can't understand behavioral aspects. In a mouse or animal model, you can understand the behavior, but then they don't inherently get autism. So,
but because we are generating these organoids from patient samples, and the cells now that we culture outside don't have the microenvironment that they would have when they're in the body. So are the characteristics of the cells similar to how it would be if it is part of our body? Or like in the sense, like there are studies that show that a person who is suffering from autism, the brain size is typically bigger than a normal human per se. But if we grow the organoids and compare them with a controlled organoid, is the size of the autism organoid bigger than the normal?
Yes, they have shown this in studies. So a lot of the what we can do is, once we generate these models in the dish or in our labs, what we have the opportunity to do is compare it to, kindly enough, to people who have donated post-mortem brains. Not enough, but there are post-mortem brains, so that we know that the tissue we are generating is equivalent to an actual brain. So first is that. And the second is, does it show similar phenotypes or similar cellular molecular structure as the post-mortem brain? I know it's not an equal comparison, but it is as close as I can get to a brain without asking one of you to open your skull and do a live study. So yes, that's the closest we can get. And yes, so going back to your question, yes, people have seen overgrowth of some population of autism spectrum disorder children show increased brain volume, and that has been shown in several studies in organoids. And one of our studies that was published, and this was during my PhD, we showed aberrant neurite growth or neuronal growth or excessive neuronal growth or neurite growth, the dendrites or in colloquial terms, the wires that come out of your neuron. Those were overgrowing, and we saw those overgrowing. And that's how we go to the overgrowth of a brain and a brain-related organ. Get to that stage.
Okay, so how do you think we are moving forward, or what is the future of studies in autism?
The future of studies, as I said, the more we get patient cell lines and patients that volunteer and ask their doctors for stem cell related precision or personalized medicine concepts, the more we can get people to donate their skin cells or blood cells, and we can convert them into stem cells. And once we do that, the more we generate these brain related cells, we can get to a point where there are enough patients in our banks that we can now analyze their cellular molecular development. And if there are patterns to that cellular molecular development, let's say there's about 100 patients that we analyzed, their brain development, and those 100 patients have a particular phenotype that they show, and we can cohort them. And we can say, okay, this patient with social awkwardness shows developmental pattern X, which is bad in the cells, and then we show that to the doctor, and the doctor can see it and say, okay, now this patient also has social awkwardness. It has the same diagnostic criteria as those 100 patients, and in those 100 patients, drug YZ, whatever it is that is approved in the market, works for those 100 patients, then I can give it back to this guy or this patient that came in the clinic. So I don't even need their stem cells anymore. If we have enough of these patient banks and stem cell banks and organoid banks, we can get to the point where we can train computers to cohort or divide the patient populations to find drugs that work for patient population A, patient population B, patient population C, D, E, F, whatever it is. And then the doctor can give that drug to that patient. This is what I hope to see in the next 5 to 10 years for autism spectrum disorder. And that's the easiest one to get patient samples for, because kids are very happy and they always are happy to talk and donate samples. So
thank you, Dr. Kathuria, for all this information, and I hope one day we do find a solution to solve these or find a solution for these disorders. Do you have any final overview or any comments for students who are just starting to understand what these diseases or disorders are?
I would say if you are intrigued about neuroscience and the brain and the mystery, we haven't even covered barely 1 to 2% of what we can understand of the brain. So if you are a budding neuroscientist, neuroengineer, biomedical engineer, medical student, whoever you are, and you are interested in understanding the brain, you have a lot of opportunity. And if you need to ask any questions, we're always here to answer. So that's what I would say. Whether it's autism spectrum disorder or just learning how the brain functions, just the basics, again, we have a lot of better understanding than we had 20 years ago, but we have a long way to go. And with that, I would ask the audience to send us questions, especially young budding students. And if they need career advice, we're always there to help you with that as well. And that's it for this podcast. Next podcast, we'll work on another neurodevelopmental disorder. Any particular favorites, Ananya? We do a lot of those.
You'd probably focus on schizophrenia.
Okay, schizophrenia it is. Next time we'll stay tuned for schizophrenia.
Thank you.
Thank you.